All right, good evening, everyone, and welcome to the MedSchool Mentor Leaders in Medicine webinar series. My name is Jun Kim. I'm the founder of MedSchool Mentor, and today from our MedSchool Mentor team, we have Matthews, Carlota, Gustavo, and Akshay joining us. And we have the incredible privilege to be joined by Dr. Chetan Bettegowda, the Harvey Cushing Professor and Director of Neurosurgery at the Johns Hopkins University School of Medicine. Dr. Bettegowda is an internationally recognized as a leading neurosurgeon scientist, bridging clinical excellence and groundbreaking discoveries. His clinical practice focuses on the surgical management of brain and spinal tumors with expertise in primary brain tumors such as gliomas, meningiomas, and pituitary tumors. In the research realm, he has pioneered advances in circulating tumor DNA as a biomarker for early cancer detection and precision medicine, helping to transform how we diagnose and monitor brain cancer. Aside from his clinical and research, Dr. Bettegowda is also widely respected as a mentor and leader in academic medicine, shaping the next generation of physician scientists and advancing the field of neuro-oncology worldwide. So, Dr. Bettegowda, thank you so much for joining us today. I believe you have a little presentation for us. The stage is all yours.
Oh, thank you, June. Overly kind and generous introduction. It's a real privilege to be here with all of you, and thank you all for taking the time to meet with me today. I will share my screen here. Is this, June, looking okay for you?
Yeah. Yes.
Okay, perfect, perfect. Thank you, team. So, I've just got a few slides that hopefully will reflect on my journey here at Hopkins and even before, and really a theme that to me has been incredibly impactful, and that's the idea of teamwork and mentorship. And for me, any of the successes that I've had personally or professionally or because of the incredible people I've been around, the incredible people I've learned from, and the groups and teams in which I've had the privilege to participate in. And currently, as chair or director of the Department of Neurosurgery at Hopkins, I feel like that's the greatest privilege that I've been gifted thus far, is to have a wonderful group of teammates who are dedicated to improving the lives of patients and the families affected by neurosurgical disorders. So that's really the theme of my slides. And I'll start with just a little bit about myself. I was born in India, in a small town, the southern part of India called Mysore. And this is actually pictures from the village in which my father grew up. And these are actually pictures from December of last year, so less than a year ago, of what that upbringing looked like for my father and grandfather and generations before them as farmers in rural India. And not much has changed over the lifetime that I've visited, and it's a special place for me. And this really speaks to, the power of really working and being focused in one's goals. And my parents, like many in the United States, immigrated to sort of give opportunity for a better life and education for my brother and I. And so we moved to North Carolina when I was 6 and my brother was 9. And I grew up in North Carolina, went to undergraduate there as well. And this is a picture of the Duke University Chapel where I completed my undergraduate degree. And it's a wonderful 4 years. And it's a picture of one of my early mentors. The theme of mentorship for me started early, and certainly I had them even in high school. But I would say one of the grounding figures of my educational life was at Duke when I met this gentleman, Thomas McCullough, who is a professor of religion and since retired, of course. But when I was at Duke, I was a major in biology and religion, and people always ask me, "Wow, that's a strange combination. Why those two?" And for me, it was a natural combination. At that time, I wanted to understand what it is that I want to accomplish in my life and try to understand how to lead a purposeful and meaningful life, contributing to others and not just oneself. And so to me, I took a lot of philosophy and ethics classes and thought about religion. And Dr. McCullough was my thesis mentor and taught me a lot about sort of ethics and morality and how to sort of live life. And so with that, I had a deep passion for science, understanding how to use science to help others led me down the path of medicine, knowing my family as a doctor. And so kind of through exploration and experience at Duke led me to medicine, and was fortunate to get accepted to Hopkins Medical School, and have been at Hopkins really ever since. And so since age 22, I came to Hopkins, and it's become my second home. I owe a lot to Hopkins, including where I met my wife, and we've had two boys, ages um, 14 and 16 now, and so really owe a lot to this university and to this institution. And they introduced me to people like Liz Jaffe and Anthony Rosen and Bill Nelson and Don Coffey, who has unfortunately since passed, but folks who in that time of their careers were fairly early. And these are all, for folks who follow Hopkins medicine are real giants in the field now, but they were early in their careers, and they introduced me to this idea of being a clinician scientist. Not having had role models in my family in medicine, I thought being a clinician was about taking care of patients, and I always assumed I'd go back to North Carolina, open up a clinic and take care of patients because I thought that's what medicine was about. But these folks introduced me to this idea of being a physician scientist, that one can see patients, do investigative work, and push the field forward. And I got enthralled with that. I applied for the MD PhD program after my first year, joined the MSTP program at Hopkins, and really was fortunate to work in this laboratory. And this is, for me, perhaps one of the most powerful examples of mentorship for me as an individual. Gentleman on the right is a person named Bert Vogelstein. And the person on the right, sorry, person on the left is Bert Vogelstein. Person on the right is Ken Kinsler, and they are two of the most prolific scientists ever known. Bert is known to be the most highly cited scientist in biological realm of all time, and Ken is not far behind. And so I was fortunate to complete my PhD with them and really understand nuances of doing great science at cutting edge work, leading technologies, but moreover, what does it mean to ask and try to answer important questions? What does it mean to try to move the field forward and be single-minded and focused while working as part of an incredible team? And I've had the privilege of continuing to be a part of this lab, and this is a larger Ludwig lab, and went on to do my postdoctoral fellowship, and have been fortunate to be one of the principal investigators in this lab. And so from student to now a mentor for other learners in our lab has been one of the greatest privileges. And through this dynamic team, we've been able to make really incredible advances in science that we think have helped push the field forward and impact people. I'll just give you a couple examples of what we've worked on over the years. And and before I get to that, I completed my PhD and fell in love with neurosurgery, did my neurosurgery residency at Hopkins, and had incredible surgical mentors. And this is Don Long, who was the former chair of neurosurgery, and Henry Brown, the immediate past chair of neurosurgery, who stepped down from that post in May of this year, and two luminaries, legends of the field, giants who have shaped the space, and for me personally have been incredibly supportive mentors. Zia Kazin, who was on faculty when I was a resident, is the chair at Brown. George Shallow, who's also on the faculty, Daniele Rigamonti, and Hugh Defoe over in France. I mean, these are all folks I spent tremendous time learning different things from, and really, the love and joy of being in the operating room and helping patients through surgical excellence was taught to me by these folks. And so I am deeply indebted for their mentorship on the clinical side. And so through this collaboration, we know that as neurosurgeons, this is where our work is. This is what we do daily, but that's not where it ends. We take care of patients, we have gratifying results, but all too often those results are not as good as we want them to be. And so, collaborative science, working with clinicians, working with others to try to answer simple questions that we hope can have profound impact. And so this is one of the questions we've worked on. And so this is in a collaborative way, both through the clinical folks I work with, as well as the research folks. And that's how do we understand cancer genetics to help folks who have CNS cancers? Really, this path has led me to think about and study tumors and processes that I never thought I would talk about or think about, and rare tumors like PXAs, or common tumors like GBM, unusual syndromes that lead to cancers like malignant peripheral nerve sheath tumors and ependymomas and chordomas throughout the CNS. And this is through the wonderful collaborations that I've had at Hopkins and other institutions around the country and globe. One of these was an early project that I worked on as a postdoctoral fellow, looking at this tumor type, which is an oligodendroglioma, a form of brain tumor that's common in young people. It has this classic fried egg appearance on histology. And we knew that it's characterized by this very specific change in the DNA chromosomes called the 1p19q co-deletion. This was known to be an unbalanced translocation. This has been known for many years. What was not known was why this is happening. And so through collaborative efforts, we're able to publish this paper in Science several years ago that showed that it really is thought to be a mechanism for inactivating two genes that previously had not been implicated in brain cancer or brain tumors, and one on the chromosome 1p and the other gene on 19q. So, this was interesting. This has now been several years, and so where did we go with this? And this is through, again, collaborative efforts through many. So we made that interesting biological insight. It led to a cool paper, and that was great. But where else can science take us? And this was a study in 2013 that allowed us to study a gene called the H13 gene, the human telomerase gene, which we, in a collaboration with folks at Duke University, noted that was commonly mutated in brain tumors like glioblastoma and oligodendroglioma. And in fact, it's the most commonly mutated gene in those tumor types. And so where do we go from there? Again, in multidisciplinary fashion. We put out this small paper in 2012 that suggested that really the genetics is what we should be using to help identify molecular subtypes of gliomas. It shouldn't necessarily be just resigned to what they look like under a microscope in the histology. And we thought really, depending on what the mutations are, you get a different lineage. So if you had IDH mutations along with CIC and FUBP1, you get an oligo. If you get the IDH and P53, it's an astrocytoma, et cetera, et cetera. And what we now know, and has been proven quite definitively, is actually something that mimics this quite closely. And this is through large prospective studies published in great papers in journals like the New England Journal of Medicine, that we now know that we classify most gliomas by the presence of the TERT promoter mutation, whether it has the 1p19q co-deletion, and whether it has the IDH mutation. I mean, that's really the hallmark of how we molecularly classify gliomas. And you can say, gosh, we played a role in that. That's kind of neat that we're helping to classify tumors. What else can science do? What else can we do as neurosurgeons? Well, we can start to think about ways to use these alterations to prognosticate how well patients do. And so this was a multidisciplinary study across multiple sites that showed that indeed, if we look at a specific locus, you can identify that mutations in different genes and different loci actually can help you prognosticate how well patients do. And this is in chordoma, a tumor type for which we didn't really have many molecular prognostic signs. And now we have more and more coming through. So now you can study the biology to tell patients how well that disease is going to respond and how aggressive it might be. So, okay, well, that's interesting. Prognostication is great. And then where do we go from there? So this was a little later. after that paper, we started to look at this syndrome called neurofibromatosis, which many of you know is there's type 1 and type 2. Type 1 is commonly associated with neurofibromas, a small fraction of which will develop into a malignant peripheral nerve sheath tumor. And so we were amongst the groups that were able to identify key genetic alterations occurring in the epigenetic pathway called the PRC2 complex that are the triggers genetically to help a benign neurofibroma convert into a malignant sarcoma. And so again, great colleagues at Hopkins that enabled this, including Alan Brillsberg, a neurosurgeon in our department. And what was interesting is that it was linked that the genes, one of the genes implicated was linked right next to the NF1 gene. And so this was the first evidence of what we thought was tandem genes being implicated in the genesis of a malignancy. So this led to an interesting paper that we were fortunate to publish in Nature Genetics. But more importantly, you fast forward several years, you're able to get science and grants published, papers published. But more importantly, now in 2024, you have folks like at MSK doing clinical trials, studying drugs that target these pathways that were found to be altered in papers that were published, 9, 10 years prior. So now you have clinical trials being enabled by biological discovery and science that was done in a team-based approach. What else can we do? Well, this is something that June talked about, which is our interest in understanding circulating tumor DNA and liquid biopsies. And won't belabor the point, but the idea is that tumors shed certain molecules into circulation and cerebrospinal fluid, and this is normal. It's part of cell turnover and cell death. Normal cells do it, but cancer cells also do it. So because we're a group that's really focused on cancer genetics, thought that this could be a way of understanding what's happening in the tumor without having to actually do surgery. One of our challenges as neurosurgeons, of course, is that it is relatively invasive. We can't just routinely biopsy things in the outpatient setting like we could for a skin lesion, for example. And so the thought was, how do we do this for folks who have cancer? And we started by looking at blood tests. So this was an early paper that we looked at saying, gosh, what fraction of cancers that we study shed detectable levels of these tumor DNA molecules in the bloodstream? And we saw this early graph, and we saw that brain cancers and gliomas were at the bottom end, 10%. So this is biology speaking to us, and now science has evolved, and we can get into that if folks are interested, but we as a group thought, why don't we look at other ancillary biofluids? And we thought about using cerebrospinal fluid. That's something we as neurosurgeons are very familiar with. We know that it bathes the brain and should be enriched for molecules of DNA coming from a tumor. And indeed, we saw that. And through collaborative efforts, we were able to develop an assay that can be used to actually help identify disease in the brain. So these are just case reports, uh, 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 just one example. This is a patient on the left who presented to clinic with signs and symptoms of leptomeningeal disease. The molecular test from the CSF was positive, but you can see the MRI was actually negative. You fast forward 6 weeks later, the patient's symptoms persist, get a repeat scan, you now see signs of leptomeningeal disease. So evidence that perhaps a molecular test may precede radiographic change. And I'll sort of end with this, that after many, many years of collaboration, publication, work by many groups, and I certainly do not want anyone to walk away from this talk thinking it was just our group, but many groups publishing in this space, pushing the field forward, we're able to finally actually allow these sorts of discoveries to enter into our guidelines. So what is it that we use to, in part, help us understand what the standard of care is to take care of folks who have cancers in the US? Well, one of them is this organization, the National Comprehensive Cancer Network. They generate a set of guidelines that are used to sort of establish what standard of care looks like, and they have it for different groups of diseases. And so this is just one example, and it's now been incorporated into other diseases like medulloblastoma, ependymoma, but I'll just share one example that this is from earlier this year, 2025, for leptomeningeal metastases, the scenario we were just talking about. They now have this little footnote that says that the assessment of cerebrospinal fluid tumor DNA increases the sensitivity. It should be used when we're suspecting there's a LMD or we're trying to use a biomarker to help track this. And so now, we go from 2014, where we first started this, to 2025, that these technologies are entering clinical space. They're changing guidelines. They're being used in clinical trials. And some of the biological discoveries are now used to not just impact biology, but being used for things like prognostication, classification, therapeutic discovery. And so, I think that I'll end here and just say that to the group and to the learners in the audience, really, this is not about me. This is about me being privileged to be part of exceptional teams, great group of individuals who believe that we can make a difference, and that together, and by sharing minds, thoughts, and ideas, and by persisting over many years, that we can actually push the field forward. And I would say that to all of you, as you think about your embarkment onto your own personal careers and journeys, I would just leave a few themes. That number one, identify mentors that really are going to support you and you can work closely with. Number two, work in groups and teams. Some things can be done individually, but most things in medicine, and specifically neurosurgery, I think are better done as teams and in collaborative spirit. I think that's really key. Number three, I would just say, find questions that you find deeply meaningful and passionate. I was trained scientifically as a cancer geneticist, so you can see all the examples I showed you today are leaning into that direction of cancer biology and cancer genetics, but that does not mean that's the right answer for everyone. There are so many ways to make impacts as neurosurgeons, as clinicians, as folks who take care of patients. Sometimes it's in the laboratory, like I've been fortunate to do. Sometimes it's through clinical trials. Sometimes it's through surgical innovation, education, public health, whatever it is that sort of is most exciting, most thrilling for you, follow that dream. I think that's really important as well. So I'll stop here and open it up to questions. And thank you again for the opportunity.
Thank you a lot, Dr. Bettegowda. After watching your slides, it really stuck with me when you said that back then you thought you were just opening a clinic and treating patients, and it was only after that you fell in love with research. And I kind of reminded myself, because I started a few years ago enjoying research and getting involved in research, and was only this year, back in January, when I did an observership in the Brigham and Women's Hospital, that I entered the first time in a lab. It was my first time seeing a lab, and it blew my mind. It was amazing. I met a guy named Junkin, and he told me about oncolytic viral therapy, and he explained to me everything about it. And
Yeah.
it just blew my mind. So I wanted to ask you, did you have a moment like this that you got mind blown when this occurred?
Yeah, great question. So for me, it really happened in first year of medical school. I listed just a few names of folks who were clinicians, but were also exceptional scientists that really came to lecture us as first year medical students. And I was so fortunate that really in every block, in every section, there was additional faculty, not just in cancer, but in all aspects of medicine, who came and talked about their patients and then said, gosh, this is the problem. And this is the science as we understand it behind the problem, but our understanding of that science is not enough. And so honestly, my first, I would say, quarter, 3, 4 months of medical school was just like, right? Like, oh my gosh, this is incredible. This is an amazing clinician who is running trials or in the lab or doing science. And I said, man, what is this? How do they do this? And so I started meeting with them after class, asking them, really directed questions like, how can I do this? What if I want to be part of this process? What is it that one needs to do? And there's no one answer that works for everyone. For me and the way I learn, the way I think, I really need sort of dedicated time and rigor to focus and get the skills and feel really good that I'm trying to do something at the highest level I'm capable of. And so that meant for me taking the time to switch into the MD PhD program, getting a PhD, spending those dedicated years. But I understand that's not the right choice for everyone. You certainly do not need to do a PhD to do great investigative work. But knowing myself and how my mind operates, I knew that level of sort of structure and rigor was important. So that's sort of how it came about for me.
Thank you, sir.
Dr. Bettegowda, thank you so much again for being with us. And talking about mentorship, I know that one of your mentor was Dr. Levy, who is also my professor at Gemelli, and he's a really important mentor and figure for me. He also showed us a picture of his portrait, I guess in your lab. Not sure about that, I don't remember. My question is, how can we, as a young medical student or physician or resident, recognize a good mentor, and also how to become good mentees for him?
Yeah, no, great question, Carlota. And when you see Dr. Levy, please give him my best. He's
Yeah.
an absolute gem of a person and has been so instrumental to me personally and professionally. And indeed, we have a picture of him in one of our conference rooms in our
Okay.
neurosurgery floor. So he's an amazing person. So he embodies a lot of what it is to be a great mentor, and one of the things to understand is that a mentor-mentee relationship is a two-way street, meaning that both parties have to commit to it. If it's just a mentor committing energy, but the mentee is not interested, or vice versa, the mentee is committing a lot of time and effort, but the mentor is vacant or absent, then that relationship is never going to build. So I think the characteristics that I've always found helpful in identifying mentors, and I have plenty of mentors even today. So it's not like one gets to a stage in one's professional career that mentorship is no longer important. I think that's not the case, at least for me. Mentorship is continually something that I seek and need. I think, someone who values what you are interested in doing, someone who is transparent and has sort of the same philosophical aspirations that you're in. I think it's helpful to have that connection both on a personal and professional level to be able to row in the same direction, identify goals, work together to achieve them. So I think identifying that kind of a person sometimes can be take a little time to identify, especially a new place, if someone's going for a new place for medical school or residency, but definitely meeting with different people, understanding what style of mentorship one needs is important. For mentee, I think it's good to have really open conversations and honest discussions about what are the goals and objectives of this, collaboration between the mentor and mentee. What is it that one would like to gain? Is it skills? Is it exposure? Is it ideas? Is it connections? Is it a paper? Is it networking? What is it that we're trying to gain, and how can we together achieve that? And I think for the mentee to sort of be able to have that crystallized in their mind, for me as a mentor, it's helpful because then I can help together chart a path to be able to achieve those goals. If it's very nebulous, that's okay. It's a good place to start, but at some point it should, I think, start to crystallize and become more clear. So I think the other piece of it is obviously, both groups need to be dedicated to the goals that they're working towards. So if the mentee can really show that they're interested, engaged, enthusiastic, I think that goes a long ways to enhancing that relationship.
Okay, thank you so much.
Sure.
Thank you, Dr. Bettegowda. Just piggybacking on Carly's question. So if you could go back in time when you were a medical student, let's say first year, second year, or third year. I'm a third year medical student, so kind of asking that question, how would you go about or recommend medical students and residents, how should they go about in their clinical rotations, in their preclinical years? How can they position themselves well to be able to learn the right things so they can succeed?
Yeah,
Thank you,
yeah,
Jim.
thanks, Akshay. So I didn't know that I was going to be a neurosurgeon coming into medical school. I was pretty open to everything, and so I think that even if one knows what they want to do deep down, I would approach each course, each piece of information, each section in first and second year of medical school, as well as all the rotations, as if this is the field that you're destined to choose, and this is what you're going to spend the next part of your life really studying deeply and practicing. And I think that way you can sort of experience to the best of your ability what that would look like. Understanding the science early in medical school, and then of course going on to the wards and doing the rotations, seeing what that looks like in practice. I think those would all be really important. And the reality is that the more we know, I'm a neurosurgeon, we practice only a very small aspect of medicine, but the more we know about other arenas of medicine, the better we can be as a clinician, the better we can be as a surgeon. So even though I don't take care of 99.9% of the medical problems that people might have, having understanding of that will only help me be a better neurosurgeon. And if people choose a different field, of course, help them. So I think number one, it helps people make the best choice possible as a medical student, that they know the field they're picking is the right field. Number two, at the end of the day, it's not like, oh my gosh, why did I study for that test? I'm never going to use it. It's just, countless hours of waste of time. No, I think it's actually the contrary. Even though you may forget a lot of it, there will be some baseline understanding that you'll say, oh my gosh, of course, that's what's happening with my patient, or it's going to help their family. And so I never underestimate the importance of sort of cross-fertilization of learning and ideas from different fields to bring forward to the specialty that you eventually choose. So I would say just keep an open mind, embrace each experience, really throw yourself in the mix. And who knows, you may end up loving something that you previously thought was not a calling for you. And that would be pretty neat.
Thank you.
Sure.
Then we have a question from the audience. Salmon, go ahead.
Hello?
Yes, we can hear
Hello.
you.
Yeah, can you hear me?
Yes.
Yeah, so my question is like I'm an IMG from Pakistan, and so during a clerkship, so we are mostly focused on our clinical skills because it's a resource limited place. So like I do want to apply for a research position. So what are the skills I should really be focusing on, which would help me be better as a for the program and the community? Like what should be I focus on besides that? That's my question is like, should I? Yeah, that's it.
Salma, I just want to make sure that I understood the question. So the question is, as a international medical graduate, what sorts of things should one focus on to be able to do a residency? Is that the question?
No, my question is uh, for research positions. Uh, like I want to be part of research programs. So like in a research limited state, mostly focusing on the clinical aspect, they don't really come toward the research side. So I am kind of lacking on skills which a complete system would have. So what should I be knowing which I do not and can focus on my own time? So that's what I'm really asking. What are those skills?
Oh, got it. Got it. Yes. Thank you for clarifying that. Yeah. So yes, for those who are in an environment where gaining research skills are not as accessible, not as prevalent, what can one do to gain those? And I think one of the nice things nowadays is that there are online tutorials and even free courses that one can enroll in to learn about, some of the basics of biomedical research, whether it's, how to ask hypotheses, whether it's one is interested, say, in big data and computational biology, taking courses on that, or listening to webinars or podcasts or TED Talks on the areas that are of interest to you. And it's impossible for any one of us to master all the areas of medicine or all the areas of research. And so what I would suggest, Salman, is to identify one or two areas of research that you think might be interesting and really probe and explore it. And with the online resources, many of which are, free or nominal in expense, to really use that. And that actually is recognized by people, that if one has sort of gotten exposure, I think that that's helpful. And then it can also help one refine what you want to do going forward based on those initial experiences.
I do have a follow-up question. Like, am I audible?
Yes.
Yeah. Yeah. I basically I study at King Edward Medical University, so it's a very high turnover hospital. Like, you do get a lot of patients every day, but as I said, it's again clinical focus. So what can I use? Because this is not all people have an access to such hospitals, like right there. It's my home institute. So what can I use that to, towards, like, basically I'm asking how would you mentor me to use that environment to improve my skills in research?
Yeah, yeah. So that's a great question, using the home environment to be able to ask questions. What I've always asked folks to do, students, residents, fellows who work with me, is to just keep a notepad of things that you see, especially in a high volume, high turnover clinical setting, of things that you see that are just perplexing. Why is it that we can't take better care of this person? Why is it we don't have an answer to this? Why is it that we have to do this in order to answer such a simple question? What led me down the path of trying to develop biomarkers for cancer is that it's perplexing to me in neurosurgery that we had to open up someone's skull to do a biopsy to understand what was happening in the brain. I thought, gosh, it's really invasive. Why can't we find other ways? And so, and so I still do this. I do take notes of things that I think we need to do better. And it can be anything. It can be in science. It can be in efficiency. It can be in safety. It can be in health economics. I mean, all of these are really important. And when one is in an environment where there are a lot of patients and a lot of sort of turnover, it's, I think, easier to think about clinical questions that one says, gosh, that's really something we should do better at. Write that down, read about it, think about strategies, and then hopefully work with folks who can be mentors to try to answer and address those shortcomings.
Dr. Bettegowda, thanks for the opportunity. Your presentation has only made me even more interested in working with neuro-oncology in the long run. Looking at the future of neurosurgery globally, what innovations, whether in surgical techniques, imaging, or molecular diagnostics, do you believe will most redefine how we treat patients in the next 10 to 20 years?
Oh, well, first, Gustavo, so glad that you're interested in neuro-oncology. All fields of medicine have needs, and for me, seeing patients in this space, I know how big that need is, and we need talented young people like yourself to fight that battle. I think that all of those things that you're talking about, whether it's molecular, whether it's imaging, whether it's engineering, whether it's AI, I think all of those are impacting medicine, specifically neuro-oncology, in huge ways. I think we're already so much better at understanding things in the last 10 years. I think in terms of surgical innovation, I think we're going to be able to do things even more and more minimally invasive. I think we're going to be able to have tools that are going to allow treatments for disorders that currently we have to do surgery for. My sense is in 10 to 15 years, rather than trying to treat something, we can actually deliver something. Instead of removing it, we can give a drug or a medicine or a gene therapy or cell therapy. That's really what I'm super excited about in neuro-oncology, is this idea that, I would love to put myself out of business, that I no longer need to take out a brain tumor because we have so many effective non-invasive strategies like drugs or, antibodies or other things. Like, I'll gladly pivot and find another career to focus on if we've done that. And so I say that a little bit facetiously, but also truthfully. But in the next 10 to 15 years, I do think we're going to be doing things less invasively and then hopefully taking things away from the surgical realm to, the medical or technology realm and not have to put people through invasive things like brain surgery, spine surgery.
Future looks bright, isn't it?
Yes, absolutely.
So, Dr. Bettegowda, you talked earlier about Dr. Bram, and we have the privilege of having Dr. Bram with us earlier this year. And in terms of cultivating a strong department, what was the most impactful lesson you learned during the brain era that resonated with you?
Yeah, Dr. Bram is such a great friend and mentor. I owe so much of my success to him, and we're so privileged that he's still on our faculty and contributing in incredible ways. And, I think one of the things that he has done incredibly well is this idea of family and culture within our department. This idea that we are all together, a part of something that is really meaningful, and we're working as a group in deep and rich ways. And it's this with sense of pride and purpose, but also in the sense of kinship and friendship and fellowship that, we are this neurosurgery family, the way he's always treated us and respected us and encouraged us in our own personal lives to do the things that are important to us, despite busy professional lives. And he has modeled that as well as any person I've ever seen. And I think that sense of kinship and family within the department, I think it's special. I'm so grateful that he has established that here. Yeah, incredible.
And I have a follow-up on that. Building on that, in your view, what role does a department chair play in shaping the culture and success of the department? And what qualities or practice do you focus on to fulfill that role effectively?
Yeah, I think that it's another wonderful question. I view one of the central goals as department chair or director to really enable people to achieve their highest and best potential. And it's not in a way that's dictated by me or someone else. I think that faculty, students, learners, fellows, they need to sort of design for themselves what that future looks like. What is it that they're deeply passionate about? And as department director, my goal is to help enable that. And for each person, it should look different because no two people should have the exact same, set of goals and interests. So that's number one is I think, being there to remove obstacles and to enable people to reach their highest potential. I think the second thing is to be able to establish, as I mentioned about Dr. Brown, a culture that's meaningful, that's allows people to come to work and find fulfillment and happiness and joy in what they do. And that's got to be steeped in things like respect, collaboration, decency, ethics, honesty, integrity. I mean, those core values. And I think it's important for folks to know that that's valued and that's coming from leadership, and that I will hold them accountable and they should hold me accountable for delivering on those sorts of key attributes and traits. And so I think helping establish sort of the mission and values of an organization are also important for the department director to play a vital role in.
Thank you, sir.
Sure.
Dr. Bettegowda, I have a question about basic science research. Because in countries like Italy, we are not really exposed to the lab or to basic science research in med school or even high school. So if we want to apply for a postdoc position that involved also basic science research, but we don't yet have the experience, what advice would you give us on how to bridge this gap before applying for this position? And also, in your view, of course, how is basic science research valued compared to clinical research when we have to apply as candidate for a neurosurgery residency position?
Yeah, yeah. These are tough questions. You're right. You're asking, really how can students sort of break into a field
Yeah.
if they don't have experience in that specific area? And, I think one of the things is what we talked about earlier as a group, which is mentorship, how to identify good mentors. And the goal is to be able to ask mentors who know you really well to help facilitate introductions to say, gosh, I know person X, they're really interested in this. Is that something that you would like to learn more about? And then have that relationship between mentor and the group that you wish to work with help facilitate a conversation to have this wonderful mentee or wonderful student who doesn't come with many years of experience, but you know what, is a great person, is industrious, is hardworking, creative, all characteristics that will allow one to succeed even in basic research. And so I think that would be a great way to make that introduction. I think another way to do it is to really, if one is very interested, dedicate time to it. Like say, I'm willing to sacrifice X number of months, years even, to gain skills. And I think that resonates with people who say, oh, this is a person that's really committed to learning this at the highest level, more than just at a cursory level, more than just an exposure, more than just say spending a week or two in the lab. That's a short window to be able to learn something. So also being able to put oneself out there and say, I'm really dedicated to this. I don't know much yet, but we all started without experience at some point. That's the whole thing about experience. Until you have it, you don't. And so we all started with nothing. And so people get that, whether it's with basic research or clinical research, it's the same. We all start from zero and gain from that. I would say that in terms of getting into a residency, what we look for, and I think many centers look for, is not necessarily a deep interest in basic lab research or deep interest in clinical outcomes research or a specific niche. No, I think it's more this idea that this applicant is very much interested in pushing the field forward. It's very interested in asking important questions and trying to answer them. And that can be in the clinical realm, it can be in the science realm, it can be anywhere. But to do it in a thoughtful and methodical way, trying to help the field and pushing it forward, I think to me that's more important than, hey, this person's really a scientist, or hey, this person's really a clinical investigator. I think we need all of it. And so I think that it should be what drives you individually as interesting, and then really dedicating oneself to that I would suggest.
Thank you, because we always like have different opinion and point of view, so it's kind of hard coming up with a like final answer or what we have to do. So
Yeah, I would say the first North Star, the guiding light would be, what is it that you're most excited about? And really go at it and delve into it and study it deeply. I think that will show and shine.
thank you.
Sure.
I can take the next question. My question for you is, with the advancements in the AI and the need for research in the field of neuro-oncology and all the technology that's been coming up, is there any idea you've been brainstorming about that you may pursue a research question in?
Yeah.
Like something exciting, something cool you've been working on?
Yeah, there's a lot of great stuff happening in the AI and digital space. One of the things actually later this week that I want to that I've been talking to one of the medical students taking a year off working with me on is, how do we use AI to help inform decision making for really rare disorders? So neurosurgery, especially neuro-oncology in general, these would be rare conditions, only a few thousand gliomas a year in the United States, for example, infrequent. And then you have all the subtypes. And so how do we understand how to take care of people if there's 500 spinal cord tumors a year in the United States? How do you understand that? Can we use AI and sort of technology to help predict what people how people are going to do, what they might respond to, what they're going to be resistant to, which patients should go on clinical trials. And so this idea of using data to help guide clinical decision making, I think is something that generally I find super exciting. And I think we're going to be using more and more, and something that I'm personally working on in the research realm as well.
Thank you.
Sure.
Dr. Lenz, a follow-up question. Much of neurosurgery worldwide is still practiced in resource-limited settings. I'm currently based in Brazil, and I do hope we'll be able to solve these kinds of issues here in the future. And if you had to design one scalable innovation to improve global access to safe neurosurgery, what would it be?
Oof. Yeah, this one I gotta think a little bit, Gustavo. That's a great question. So one innovation to increase safety for neurosurgery globally, is that the question? Yeah, yeah. I'd love to be able to have some sort of an online um expert panel that we could all access, where somehow we have it so that we can share in HIPAA compliant ways medical information and remove any of the legal ramifications of saying, oh, someone's practicing outside of their country, or someone is, this is malpractice and we're gonna have liability if we do this. So let's say we can remove all those logistical hurdles. I would love to see, just a panel of experts from around the globe. And if I have a complex case, I can go to this group and say, hey, what does this group think? There's some extent of this, there are virtual tumor boards and other things that approximate this, but I think really for neurosurgery, we need more than that. We need intense dialogue, we need review of the imaging and clinical features, and then a discussion of the pros and cons of different approaches. And so, if it's truly a complex situation, it's rarely, oh, I would just do this, and that's the right answer. It's usually a discussion. So having an opportunity to do that globally, I think would be amazing. How do you resource that? How do you get people who have time to participate in those roles? I think those are some of the logistical hurdles, but technologically we have all that. It's not like we need something new to be discovered to enable it. It's just, I think creating that infrastructure would be really amazing. Yeah.
Sounds good to me. Thank you.
So, doctor, changing subjects a little here. You are a very high achiever, and in addition to your neurosurgery career, you are also a husband and a father of two. So, how do you balance family life with high intensity demands of being a department chair, surgeon, and scientist?
Yeah, I think that's the most important question that I ask on a regular basis. Is all of these are very important. I think obviously for me, my family is really is the priority. And for me, it's trying to be very intentional in how I spend time, and trying to immerse myself deeply in what I'm doing. So if I'm with family, really trying my best to focus on them, to be in the moment so I can appreciate and enjoy. So even if that time is shorter than I would like, it's still a really deep and rich experience, both for my family and for me. And, rather than being distracted all the time, or if I'm at work, trying to be really focused and efficient, knowing that there are many competing challenges for time, but if I'm working on something, really trying to focus on it so that I can be efficient, so that hopefully it opens up more opportunities for me to contribute to the other aspects. And so for me, it's about being super intentional in how I spend my time and trying to be efficient and focused. But the last piece of it also is that I love my professional life and I love my family life. And none of that is considered a burden or a challenge or a negative. Like, it's spending time doing things I care about deeply, super excited about. And so it's all like, gosh, let me go from really something I love here to something I care about there to, and not that I'm not saying that every aspect doesn't have challenges. Of course, life is full of challenges, but one can still have love for that part of one's life, even if it's difficult, even if it's challenging, even if it's hard. Most things in life that are meaningful are hard, but one can still love that. And so that's what I mean. I'm not trying to say my life is perfect and there's nothing difficult. No, I have the same challenges that everyone does, but I still have a lot of happiness that comes through having those interactions. And so I think, that's sort of in terms of grounding myself and not feeling burnt out or overextended. That's really, I think, important for me.
Yeah, thank you, sir, for answering.
Sure.
And then we have a question
I
from Faraya from the audience. Faraya, go ahead.
Hi, Doc. So I had a quick question for you. Well, first of all, actually, when I was listening to your family background, it really hit me because I was also the same age when my family came to the US. So I think you can probably understand that coming to the US and coming to a new country where my parents did not go to med school here, they went in their countries. So navigating a new space, it's really different from from what they're used to. So how are you, I'm a pre-med student, so how are you able to find opportunities for example, where the local institute, like med school here, does not have a lot of clinical research opportunities, and I'm in my gap year doing a master's in basic science, neuroscience research, but I also do want to get involved in at least some clinical neuroscience research, but I just don't know how to go on about it.
Yeah, yeah. So, what I would suggest is to sort of think about the fields in neuroscience, clinical neuroscience that you're interested in. What are the areas? And that's a broad, part of medicine. What are the specific areas within that are interesting to you? And then I'd say, gosh, who are the people doing the best kind of work? What is it that I'm interested in and looking through PubMed and literature, you can identify hopefully a handful of groups that are really doing great clinical neuroscience. And honestly, one of the benefits of COVID is that the whole virtual experience has gotten easier. And so, our lab group will have folks who participate remotely on very specific projects. So finding a clinical neuroscience group that is studying things that are interesting to you, and then asking and reaching out to those PIs and say, gosh, exactly as you explained, I don't have this opportunity locally, but I'm deeply committed to this. Would love to learn from you. Are there, virtual sessions that I can participate in and learn more, gain responsibility, gain trust, and then from just being a listener to being a participant in that work. And one of the good things about clinical research or clinical facing research is that it can be done remotely in many cases, depending on the type of work. Harder for bench work. A lot of times bench-based laboratory research, one physically has to go there. But I do think the clinical work can be done in conjunction through collaborative stuff online. So that would be, I think, one approach to take.
Cold email the people, because I'm really interested in I currently do glioblastoma and stroke basic science research. So there are a handful of people that I constantly read about. So would you just recommend cold emailing them?
Yeah, so one can cold email them, but it's also, an opportunity where if you have a mentor that you feel comfortable asking to facilitate an introduction, sometimes that can garner more of a response because, faculty are getting a large number of emails and we want to try to get a response. Sometimes having an introduction from a mentor can increase the chances of a positive response.
So we are out of time here. I want to thank you, Dr. Bettegowda. It was truly an honor to have you here with us. I want to say to everybody here that we have a lot of our Leaders in Medicine webinars on our YouTube channel, and this one will be posted as well on YouTube channel. So thank you all for joining us. And again, Dr. Bettegowda, thank you.
Thank you so much for the opportunity. I wish everyone the very best. Take care.
Thank you. Bye.
Get out to bed.
Thank you.
Thank you.